everyone, myself Shah from Department of Pharmacognosy at LJ Institute of Pharmacy. Welcome to the book series which is initiated by LJ Institute of Pharmacy. Don't forget to like, subscribe and share our YouTube channel Pharma Ignite. In today's lecture, we are going to study definition, history, scope and development of pharmacognosy. First of all, pharmacognosy is a Greek word. It is derived from two Greek words. One is pharmakon, second one is gignosco. Pharmakon means a drug and gignosco means to acquire knowledge of the true drugs. Now, pharmacognosy is a branch of bioscience which deals with the basic resources which are obtaining from the natural source and associated with their pharmacological actions, or it can be used as a medicaments for various diseases. Now in this pharmacognosy branch, the basically study of the crude drugs and their derivative, it's a knowledge of the different characters like sensory characters, morphological characters, microscopical characters. Now, pharmacognosy includes the study of the materials and substances which are used in a pharmacy, such as the materials like suspending agent, disintegrating agent, flavoring agents and filtering agents. Now, suspending agent, the best example of suspending agent is acacia and tragacan. Now, disintegrating agent like microcrystalline cellulose, flavoring agents, all volatile oil containing drugs like fennel, coriander are used as a flavoring agent. Filtering aids, that is kisalgar, which is obtaining from the mineral source. Substances, there are various substances which comes in a pharmacognosy like antibiotics and immunomodulatory substances, pesticides and oral contraceptives and poisonous plants. The best example of antibiotic is penicillin, which is obtaining from the plant source. Then immunomodulatory agents like ashwagandha, shatavari, gado that having the immunomodulatory activity, which is used for the treatment of the various diseases. Now, the various phytoconstituent or steroids can be isolated from the plants and they can be used in a synthesis of the oral contraceptives. Now, this branch is also associated with the different branch of sciences like botany, pharmaceutics, biotechnology, then pharmacology, phytochemistry. Now, we will study about history of pharmacognosy. Nature has provided a complete storehouse of remedies to cure most of the disease. So, before Christ, herbal drug can be obtaining from the China, India, Egypt. These are the countries from where the herbal drug is first obtaining. So China widely used the plant as a sources of root drugs since 5000 BC. The oldest common herbal book is Pen Tea Sao, which is written by Shengneng scientists. In this, he includes the 365 drugs which is used for the treatment of different diseases. In a traditional system of Ayurveda, there are three components. One is Charak Samhita, second one is Shushruta Samhita, and third one is the Ayurveda system. So Charak had made 50 groups of 10 drugs or herbs which are classified on the basis of physician, while Shushruta arranged 760 herbs in a 7 districts with their common properties. And third one, Ayurveda has prescribed the various diseases or various drugs for the Tridosh system. So Tridosh system is basically divided or derived from the gunas, Vata, Pitta and Kapha. Now here Ayurveda is known as an ancient medicine of life. Now contribution of scientists into the medicine system. So Hippocrates is known as the father of medicine. He was the first natural doctor who used the natural remedies for the treatment of various diseases. Then Aristotle, Galen was the first pharmacist who had number of pain relieving materials obtained from the plant source. First, one of the best example is opium. Then other scientists' contribution, like Mary had discovered the isolation methods as well as extraction method, and he shows that alcohol is the best extractant. William Manthering shows the medicinal properties of the digitalis. Then Pelletier had isolated the strychnine from the nux vomica. Stas and Otto had discovered the different isolation methods from the alkaloids and glycosides. Then Sadler was the German scientist. He introduced the pharmacognosy in his books. The book name was Antelica Pharmacognosis. Now, various phytoconstituents, their different alkaloids are derived from the plants. Now, these are the name of the scientists who gave their contribution in a isolation of the alkaloids. Now, other important discoveries in a 20th century, the isolation of ergometrin, digoxin, reserpine can be possible, but in a 19th century, the pharmacognosy is known as the materia medica. Now, these are the name of the scientists who developed the binomial system as well as the classification of the two drugs. Microscopy is an important tool in a pharmacognosy which is used for the identification of the two drugs. So first of all, in this identification, first we need to clear the section. Second one, mounting in a different agents, and third one, we have to employ the different staining techniques for the identification of different tissues. Now, these are the name of the scientists who discovered the anatomical or powder characteristics from the plants. Now, in the 19th century, T. Search had developed the anatomical characters of several powder drugs. Now, we will talk about the scope and development. There is a wide scope of the pharmacognosy. First of all, pharmacognosy is basically used to identify and evaluate the plant parts on the basis of their evaluation parameter. Then it is an important link between the phytochemistry or pharmacology and phytochemistry, or we can say medicinal chemistry. Why? Because first of all, the drugs is available in a raw form. From that, we need to isolate the phytoconstituent. So there is a role of chemistry in the isolation of phytoconstituent, and the synthesis of the derivative from the each phytoconstituent and their uses. This synthetic or derivatives are connected with the different uses. So in a uses we need to go for the pharmacological purpose or by performing the different animal models. Now these drugs can be evaluated on the basis of physical evaluation, organoleptic evaluation, microscopic evaluation, chemical evaluation, and other evaluation methods. And now we are talking about the development. Development of the herbal drugs can occur in a different field. So first of all, we are talk about the or discuss about the phytochemistry. So first of all, the substance which can be isolated from the plants, then that structural elucidation as well as different derivatives can be synthesized. So the role of isolation method is based on a chemistry. Now second one, preparation of Ayurvedic formulation. The different Ayurvedic formulations can be prepared for the different diseases. Like the best marketed formulation is a Leaf 52, which can be synthesized by the or which can be formulated by the well-known pharma companies. So second one, there are many other examples like Shitopaladi Choda and Dashmularishta, then Sudarshan Dhanvati. So this formulation we can formulate on the basis of procedure which is given in Ayurvedic Pharmacopoeia of India. Now they are effective in a chronic disease because they having the less side effect compared to the synthetic drug, and they can be used for the long term of the treatment. Now various drugs like Vidanya, Shatavari and Gado can be used for the immunomodulatory purposes or adaptogenic without having no side effect or less side effect. See, in recent COVID pandemic situation, majority of people are using the herbal drug as the remedies. Now, there is a wide scope in a steroidal industry because the many pharmacists or industrialists are involved in a synthesis or isolation of the steroids from the plants, like diosgenin and vidanolides can be isolated from the diascoria and vidania respectively, and they are used for the synthesis of sex hormones as well as oral contraceptives. Preparation of antibiotic. In this, the well development was discovered by William Benthering. He had isolated the penicillin from the plant source. Now, various agents which are aid in a formulation of the pharmaceutical preparations. So, like for an example, saponins, which can be used as a foaming agent. The best example of foaming agents are the soap nuts, which is known as a arita, which can be used as a pharmaceutical aid as well as important ingredient in a preparation of shampoo. Second one, example glycyrrhiza. Glycyrrhiza means jetimer. Jetimer having the bitter, masking the bitter test because it is a sweetening agent. So it is used for the development of the different formulation to mask the bitter test. Then gelatin mainly used for the synthesis or for the shell preparation, soft gelatin capsule shell preparations. Fixed oil and fats are used in a preparation of ointments. Volatile oils are mainly used as a carminative or it can be used for the flavoring agents. Now biosynthetic pathway, so it is based on a chemistry. There are three pathways. Cyclamic acid pathway is used for the synthesis of amino acid. Acetate malonate pathway for fatty acid and acetate mevalonate pathway for the steroids. Now there is a wide future scope in for the herbal drugs because people are recommending more herbal drugs compared to the synthetic drug because they are comparatively safer than synthetic medicines. For an example, artemisinin and quinine is used as an anti-malarial. Taxol is used as an anti-cancer. The herbal drug having the high potency and less side effect compared to the synthetic drug. So people are recommending more usage of herbal drug compared to the synthetic drug. Now, herbal drug industry is a link between the identification of the phytoconstituent to the development of the formulation. So, first of all, the identification of the plant, then identification of the marker compound, and then this marker, by using this marker compound, the development of the formulation for the various treatments. So, during this process, the standardization will occur. So standardization is an important parameter of the quality control. Now here this is the description or I have mentioned one example in which it describes starting from the plants to the development of the product process. So first of all the natural drug opium poppy is used. Then crude drug extract is prepared. Crude drug extract means it containing the different mixtures of the phytoconstituent like morphine, codeine, thibane. So here the different mixtures of the phytoconstituent are present. Then the pure morphine can be separated out by the process and this morphine is used in the development of the pharmaceutical preparations like morphine sulfate tablets and oral solutions and others. These are the FAQs of this session. Thank you for watching my presentation.